Welcome back to the deep dive. We're jumping back into the nervous system today.
Yeah, picking right up from our last chat.
We really focused last time on the neuron, the
Uh,
actual cell firing the signals, how it sends messages, and how that myelin sheath helps speed things up.
exactly. We covered the main actor, you could say. But, well, the body's never just about one cell type, is it? There's always a whole support team.
That's exactly where we're going. We're diving into that essential supporting cast. These cells, they don't fire impulses, but honestly, neurons just can't function without them.
Oh, absolutely vital. They're called neuroglia. Sometimes people just say glial cells.
Neuroglia, okay.
And today, using Mr. Hostler's lecture notes, these are for his nursing A&P students, based on Chapter 10 of Herlihy's The Human Body in Health and Illness. We're going to unpack what they do.
Right. So the mission today, understand how these neuroglia support, protect, manage the environment for neurons, and importantly, how they make myelin.
And we'll see how that shapes the actual structure of your brain and spinal cord, too.
It's like the behind-the-scenes story.
The unsung heroes, definitely.
Okay, let's start broad then. What exactly are neuroglia? Lay the foundation for us.
Okay, so neuroglia, and you'd say new ROH glee, these are basically all the non-neuron cells in the nervous system.
Found everywhere. CNS, PNS.
Everywhere neurons are. Central nervous system, so brain and spinal cord, and the peripheral nervous system, which is all the nerves branching out. The main thing is they don't conduct electrical signals like neurons. That's not their job.
So not the messengers, they're the
Yeah.
infrastructure, the support crew.
That's perfect. They do all the background work. Think structural support, holding everything together, regulating the chemicals around the neurons, super important for them to fire properly.
Yeah.
They act like the immune system within the nervous tissue, cleaning up messes, fighting infections. Some even help with repair after injury. And, like we said, producing myelin.
Wow, okay. That's a lot of different roles. And the notes mention 6 main types.
Yep, 6 main types. 4 you find mainly in the CNS and 2 in the PNS. Knowing which is where is pretty key.
All right, let's break down the CNS4 first. Who's on the team?
Okay, top of the list. Astrocytes. The name means star cell, because that's kind of what they look like.
Star-shaped, okay.
And they do a ton. They help maintain the blood-brain barrier, that protective filter. They balance ions and neurotransmitters around the neurons. They even help with metabolic stuff, like getting energy to the neurons.
Huh. So, real multitaskers.
Mm-hmm.
Controlling the environment, helping feed the neurons.
Yeah, that sums it up well. They keep things stable, which the CNS absolutely needs. Next up, oligodendrocytes.
Oligodendrocytes.
These guys have one huge job in the CNS. They make the myelin sheath.
Okay. The myelin makers for the brain and spinal cord. How do they work?
So this is where it gets interesting and different from the PNS. One single oligodendrocyte can actually reach out and wrap segments of multiple different axons.
Wait, really? One cell myelinates bits of several different nerve fibers.
Yeah. It's efficient for the dense environment of the brain and spinal cord. Imagine one cell insulating several different wires at once.
Wow. Okay, that's a big difference to remember one oligodendrocyte, multiple axons.
Definitely. Hold on to that distinction.
Yeah.
Then we have microglia.
Microglia.
These are the immune cells of the CNS. They're small, they move around, kind of patrolling. Their job is finding trouble, injury, infection, and cleaning up debris, getting rid of damaged cells. Think of them as the brain's dedicated cleanup crew and security force.
Little janitors and guards. Got it.
Exactly. And the last one for the CNS, ependymal cells.
Ependymal. Okay.
These line the cavities inside the brain, the ventricles, and the little canal down the middle of the spinal cord. Their main gig is helping to make and circulate cerebrospinal fluid, the CSF.
Ah, the fluid that cushions everything.
Right. Cushions, transports nutrients, removes waste. Ependymal cells help manage that fluid system.
Okay, CNS team recap. Astrocytes are the support environment stars. Oligodendrocytes make myelin for multiple axons. Microglia are the immune defense. Ependymal cells handle the CSF. Makes sense. What about the PNS, the peripheral nervous system?
Right, out in the periphery, 2 main types. First, Schwann cells.
Schwann cells. Now, these also make myelin, don't they?
They absolutely do. Schwann cells make myelin, but only in the PNS. And here's that key difference we talked about with oligodendrocytes. One Schwann cell wraps itself around just one segment of one axon.
Ah, okay, so it's a one-to-one deal. If an axon needs, say, 10 segments of myelin, that takes 10 Schwann cells.
That's generally how it works, yes. One Schwann cell per myelinated segment of a single PNS axon. It's a fundamental difference in how myelin is laid down in the 2 divisions. Super important distinction.
Okay, that feels like a really critical point. Oligodendrocytes in CNS wrap multiple axons. Schwann cells in PNS wrap one segment of one axon.
Got it.
Who's the other PNS glial cell?
The other type is the satellite cell.
Satellite cells.
You find these guys clustered around the cell bodies of neurons located out in the PNS in those bundles called ganglia. Their function is kind of similar to what astrocytes do in the CNS, but on a smaller scale, they help regulate the chemical environment right around those neuron cell bodies.
So they're like the local support crew for the neuron's command center out in the PNS.
That's a good way to put it. Keeping conditions just right for those cell bodies.
Okay, we've met the 6 glial types. How does this whole support team stack up against the neurons themselves? What's the big picture difference?
Well, the absolute core difference is the action potential, the electrical signal. Neurons generate and transmit those signals. They communicate fast across synapses. They're the information processors.
Right.
Neuroglia, like we said, they just don't do that. Their entire job revolves around helping the neurons survive and function effectively, insulating them, feeding them, protecting them, cleaning up everything but sending the signal.
And what about sheer numbers? More neurons or more glially?
This always surprises people. There are way more neuroglia than neurons. In many brain areas, it's like 5 glial cells for every 1 neuron, maybe even more.
5 to 1.
Seriously.
Yeah.
So while neurons get all the attention for firing signals, the sheer amount of background work done by glia is just immense. They're the foundation.
That completely changes the picture. It's not just wires. It's this whole dense ecosystem with a massive support staff. And it sounds like myelin, made by these glial cells, is what leads to that basic division we see, gray matter and white matter.
Exactly. It's a direct result of myelination. Gray matter and white matter just describe areas based on what's mostly packed in there.
Okay, break down. What's in gray matter?
Gray matter is mainly neuron cell bodies, dendrites, and importantly, unmyelinated axons. Since it lacks that fatty myelin, it looks grayish. This is where a lot of the actual processing, the computation happens.
Makes sense. And white matter.
White matter is mostly bundles of axons that are myelinated. That myelin sheath, being fatty, gives it a whitish look. Think of these as the transmission cables, the highways carrying signals quickly between different areas.
And the way gray and white matter are arranged, it's different in the brain versus the spinal cord. That seems important.
Oh, it's hugely important, and it makes perfect sense when you think about what each part does. In the brain, the gray matter is mostly on the outside. That's the cerebral cortex, the wrinkly surface layer.
Okay.
The white matter is generally found deeper, inside, connecting different parts of that cortex and connecting the cortex to other brain regions.
And the spinal cord flips that.
Completely reversed. In the spinal cord, the white matter is on the outside, forming a thick layer around a central core of gray matter, which often looks like a butterfly or an H shape in a cross-section.
Okay, the big question then, why? Why flip the arrangement? What's the functional advantage of gray outside in the brain, but inside in the cord?
It's a really elegant example of structure-fitting function. Think about the brain's main job, especially the cortex complex, processing, thinking, awareness.
Right, higher level stuff.
So putting the processing units, the gray matter, with all the cell bodies and dendrites on the outside allows the brain to maximize its surface area.
And that's where the wrinkles come in, the folds.
Exactly. Those folds, the ridges called gyri and the grooves called sulci. They're not random. They're nature's way of cramming an absolutely massive amount of gray matter, billions of neurons, into your skull. More folds equals more surface area equals more processing power. You know, compare it to an animal like an opossum. Their brain is pretty smooth, not many folds. They just can't pack in as much cortex, which limits their complex thinking compared to us.
That comparison clicks. Maximize processing surface in the brain,
Right.
put gray matter outside, and fold it up. So what about the spinal cord setup then?
Well, what's the spinal cord's main job? It's primarily a superhighway, right? Relaying signals quickly between the brain and the body, plus handling some local reflexes.
And for fast transmission?
Myelinated axons, white matter. So putting the white matter on the outside of the spinal cord places those fast transmission tracks, those myelinated cables, in the perfect position to shuttle signals up and down rapidly. The gray matter in the center. That's where the connections for local reflexes happen, and where the cell bodies for motor neurons controlling muscles live. It handles the local processing and command outputs.
So the layout is perfectly tuned for the job. Brain processing area on the outside, spinal cord, transmission pathways on the outside. Structure follows function.
Beautifully. It's a core principle you see again and again in A&P.
This has been fantastic, really clarifying the role of the support crew and how the system is built. Let's just quickly recap the big takeaways, drawing from Mr. Hostler's points.
Okay. First, neuroglia are the essential support cells. They don't send signals, but they do everything else. Critical.
And there are 6 types. 4 in the CNS astrocytes, oligodendrocytes, microglia ependymal cells, and 2 in the PNS Schwann cells and satellite cells.
Key point. Myelin comes from oligodendrocytes in the CNS, wrapping multiple axons, and Schwann cells in the PNS, doing one segment of one axon each. Remember that difference.
Got it. Neuroglia handle structure, immunity, chemical balance, and that vital myelin.
And myelin dictates structure. Gray matter has the cell bodies, dendrites, unmyelinated bits. White matter is the myelinated axon highways.
And the layout reflects the job. Brain has gray outside, folded cortex, for processing. Spinal cord has white outside for fast transmission, gray inside for local stuff.
Understanding all that the glia, myelin, gray-white arrangement is just fundamental. They really are the unsung heroes.
Totally agree.
Right.
Which leads us perfectly to the challenge question from the notes for you, the listener, to chew on. Why is gray matter on the outside of the brain, but the inside of the spinal cord? Think about the specific functional advantage that gives each part.
Yeah, think about what each structure needs to do best. The design is all about efficiency for the task.
Mull that over. And join us next time. We'll keep building on this, looking at how the central nervous system protects itself, bone, membranes, that special fluid, and the blood-brain barrier.
Sounds good. See you for the next deep dive.